Hi everyone, I am Dr. Shiba Daniel, Department of Chemistry, Holy Cross College, Nagarkoil. Today I am going to explain about proteins, that's Introduction to Proteins. Proteins are long chains of amino acids that form the basis of all life. The human body consists of around 100 trillion cells. Each cell has thousands of different proteins. Proteins consist of amino acids, and proteins on hydrolysis by enzymes, acids or alkalis to form alpha amino acids as the final product. Two or more amino acids combine together to form peptides. Peptides containing 2 to 10 amino acids are called oligopeptides. Peptides containing more than 10 amino acid residues are termed as polypeptides. Polypeptides containing more than 100 amino acid residues is called a protein. The next is the structure of proteins. Proteins have 4 different levels of structure. They are primary structure, secondary structure, tertiary structure and quaternary structure. So first one is the primary structure. The primary structure of protein refers to the sequence of amino acids in the polypeptide chain. The primary structure is held together by peptide bonds. The two ends of the polypeptide chains are referred to as carboxy terminus and the amino terminus. Based on the nature of the free groups on each extremity. The end of the protein sequence with the free carboxyl group is called the carboxy terminus or C terminus. The end of the protein sequence with an free amino group is called the amino terminus or N terminus. The sequence of amino acids in the polypeptide chain is always read from N terminus to C terminus. Then second one is the secondary structure of protein. Linus Pauling proposed the secondary structure of proteins, and it refers to a simple folding and bending of peptide chains. The most common type of secondary structures are alpha helix and the beta pleated sheets. Both structures are held together by hydrogen bonds which form between the carboxy oxygen of one amino acid and the amino hydrogen of another. The next is the tertiary structure of protein. Tertiary structure is the three-dimensional structure and it consists of both helical and pleated sheets. The stabilizing factor of tertiary structure includes hydrogen bonding, ionic bonding, dipole-dipole interactions, van der Waals interactions and hydrophobic interactions. Next is the quaternary structure, and it is an assembly of two or more polypeptide chain which are individually folded and compacted. Each chain is the assembly is called a subunit. These subunits may or may not be identical. They are held together by hydrogen bonds and separated by dissolving in water containing urea. The stabilizing factors of quaternary structures are hydrogen bonding interactions, hydrophobic interactions, van der Waals interactions and ionic bond interactions. Here this is an example of a hemoglobin. Hemoglobin is made up of 4 subunits, 2 identical alpha chains and 2 identical beta chains. Each subunit has a tertiary structure similar to that of myoglobin. Next one is the classification of proteins. Proteins are mainly classified into 2 types: classification based on solubility and shape and classification based on chemical structure. First one is the classification based on solubility and shape. So in this classification, the proteins are divided into 2 types: fibrous proteins and globular proteins. Fibrous proteins are linear polymers which exist in the form of fibers. They are insoluble in common solvents like water but soluble in concentrated acids and alkalis. Example, fibroin and keratin. And the next one is the globular protein. They are cross-linked polymers wherein the peptide chains are folded tightly to give a spherical or globular shape of the molecule. These are water-soluble proteins. Example, hemoglobin and myoglobin. So this is the structure of hemoglobin and this is the structure of myoglobin. The next classification is based on chemical structure. Based on this chemical structure, proteins are classified into 3: simple proteins, conjugated proteins and derived proteins. First one is the simple protein. The protein which give only amino acids or their derivatives on hydrolysis are called simple proteins. Based on their solubility, simple proteins are further divided into 7 groups. Next is the conjugated proteins. These are proteins which contain a non-protein part. The non-protein part of the conjugated protein is known as the prosthetic group. Conjugated proteins on hydrolysis yield non-protein substance in addition to amino acids. That is, in addition to amino acids, prosthetic groups are also formed. Depending upon the nature of the prosthetic group, conjugated proteins are further divided into 6 groups. The next is the derived proteins. Derived proteins are intermediate products obtained by the stepwise hydrolysis of natural proteins by acids, alkalis or enzymes. Next is the properties of proteins. First one is the amphoteric character. Proteins are amphoteric in nature. So protein consists of amino acids. Amino acids consist of free NH2 group and acid group. NH2 group is the basic group and the acid group is the acidic part. So it exists as switcher ions. So in an amino acid it consists of both basic as well as acidic group, and it exists as switcher ions, and it shows amphoteric character. It acts as neither acid or nor base. The next property is the isoelectric point. Proteins have definite isoelectric point, and it depends upon the nature of amino acid present in it. The isoelectric point is the pH at which the amino acid does not migrate in an electric field. Example, isoelectric point of pepsin, casein and serum albumin are 2.7, 4.6 and 4.7 respectively. The next one is the denaturation. Proteins are optically active and are precipitated from aqueous solution by heat, addition of acids, alkalis or salt solution and loss of their activity. Proteins in the precipitated state are said to be denatured, and the process of precipitation of proteins are called denaturation. Denaturation is due to the result of changes takes place in the conformation or unfolding of the protein molecules. The reverse of denaturation is called renaturation or refolding. The next is the test for proteins. First test is the Biuret test. Alkaline solution of proteins gives a purple color when treated with the dilute copper sulfate solution. The purple color is due to the formation of a complex between copper Cu2 plus ions and peptide bonds. In this reaction, a Biuret is formed as the intermediate. And this is the Biuret test. This is the alkaline solution of the protein, and this is the violet color represents the presence of protein. The next test is the Millions test. Proteins forms a white precipitate when treated with a few drops of Millions reagent. The Millions reagent is mercuric and mercurous nitrates in nitric acid containing a little nitrous acid. The next test is the ninhydrin test. Proteins with the free amino and carboxylic acid groups are boiled with a 2% solution of ninhydrin. A purple color is developed. So this is the ninhydrin test. The next is the nitroprusside test. Proteins containing free SH, that is thiol group, gives a red color with the sodium nitroprusside in ammoniacal solution. The last test is the xanthoprotic test. Proteins containing benzene nucleus gives an yellow color when warmed with the concentrated nitric acid. The yellow color is due to the formation of xanthoprotein. Hope you understand about proteins, that is structure of proteins, classification of proteins, properties of proteins, and test for proteins. Thank you.